So the differences between DNA and RNA. DNA is more chemically stable than RNA. DNA is more durable, whereas RNA is somewhat temporary. DNA is composed of deoxyribose sugar. The pentose sugar is deoxyribose, whereas in RNA it is ribose, the pentose sugar. DNA contains thymine, and RNA contains uracil. They both contain adenine, cytosine, and guanine. But DNA contains thymine, whereas RNA contains uracil. That distinction is important and worthwhile for you to recall. Locations. DNA is found in the nucleus, in mitochondria, and in chloroplasts. Whereas in RNA, it is found wherever DNA is found because it is transcribed from DNA, but it is also traveling everywhere else within the cell, into the cytosol. So it's manufactured in the nucleus, but it travels everywhere else within the cell to be translated into protein. Now, DNA has a double-stranded structure, and RNA has a single-stranded structure. Now, there are 3 flavors of RNA, messenger, ribosomal, and transfer RNA, whereas there's just the one really big flavor of DNA, double-stranded DNA. Now, that's an assignment that we did. All right, our glossary of terms. mRNA will be back in Unit 3. Transcription. Transcription is the process of making an mRNA strand from a DNA blueprint or template. Translation is the process of making mRNA information and making a protein's primary structure. The template strand is a DNA strand that is complementary to the RNA chain that is being transcribed from. And a coding strand is a DNA strand that the completed mRNA is meant to resemble. A protein is a macromolecule that is made up of amino acids. Enzymes, more on enzymes immediately following this, but for now a gross oversimplification. It is a globular protein that has a job to do. RNTPs are ribonucleotide triphosphates. ATP is an example of this. CTP, GTP, and UTP are examples of this as well. Ribosomes, a mixture of rRNA and protein that functions as the site of translation. These are rRNA and protein. They have a large and small ribosomal subunit. A ribosomal subunit is one of 2 units, large and small, that clamp over mRNA that make up the complete ribosome. Now, this is an A-level topic that is fun for me, but not an AS-level topic. So this is how we can have activators and repressors and mediators and transcription factors that all promote the transcription of RNA from DNA. So RNA polymerase is attached to all of these transcription factors, and promoter regions are binding to these transcription factors. The transcription factors are binding to these promoter regions of DNA to help place the RNA polymerase on that region that allows for the coding of the RNA transcript. So we have enhancer regions here, we have activators and receptors that prevent the forward motion of this RNA polymerase system throughout the RNA polymerase complex down the chain to produce this RNA transcript. Now, let's talk about enzymes now. Baseline knowledge. You need to write these definitions down at home if you don't have them already in your notes. The definitions for enzymes, for the enzyme for metabolic, for anabolic, for catabolic, for catalyst, for substrate, active site, complex, globular, synthesis, denature, activation energy, pH, the bond types, covalent ionic hydrogen and hydrophobic interactions. Now, let's go through these one by one. An enzyme, as I said before, a globular protein with a job to do. This is an enzyme that has a 3D conformation. It has a specific shape and it has a specific function. That shape and that function are intrinsically linked to one another. So an enzyme performs a function. It is a protein that performs a function. Metabolic is the overall chemical reactions that a cell performs. Anabolic are the building up reactions, the reactions that build. The catabolic reactions are the reactions that break down within the cell. The net of all the anabolic and catabolic reactions is metabolism, metabolic reactions. A catalyst. An enzyme behaves as a catalyst. A catalyst is a chemical that participates in a chemical reaction and speeds up the rate of a chemical reaction, but is not consumed while forming products. So it participates in the chemical reaction, but is not consumed. So a catalyst, an enzyme is a protein catalyst for chemical reactions. The substrate is the molecule or molecules that an enzyme will act upon. The active site is the space, the physical part of an enzyme where that substrate will bond in order for that chemical reaction to proceed. A complex is the combination of multiple molecules that are not necessarily covalently bonded to one another. So an enzyme substrate complex is when an enzyme and a substrate are bound together, the substrate to the enzyme's active site, that forms an enzyme substrate complex. They are a complex, they are bound together, but they are not covalently bonded to one another necessarily. Globular. We discussed this definition before, but they are round-shaped in general, or they have a general 3D conformation. Synthesis reactions are building reactions. They synthesize, they put things together. Denature. To denature is to permanently alter the shape of an enzyme. Activation energy. An activation energy is the energy required to begin a chemical reaction. To allow a chemical reaction to proceed to completion, you must have the required activation energy. A catalyst will lower the activation energy of a chemical reaction. An enzyme is a protein catalyst, so an enzyme will lower an activation energy of a biological chemical reaction. pH. pH is the negative log base 10 of hydrogen ions. Now that is an indication of the relative acidity or basicity of a reaction. This is on a scale of 1 to 14, with 7 being neutral. All right? Low pHs are acidic pHs. That would be anywhere from 1 to below 7. Basic pHs are anywhere from above 7 to 14. The higher your number, the more basic. The lower your number, the more acidic. Your bond types. Your covalent bonds are a shared electron pair between atoms. That is your strongest bond type in an aqueous solution. Ionic bonds are a relatively weak bond type in an aqueous solution, but they are still stronger than hydrogen bonds. Ionic bonds are the attraction between oppositely charged particles. A positive charge and a negative charge will attract to one another. Hydrogen bonding is the result of a dipole interaction between a positively charged, a net positive charge of a hydrogen, for example, in the hydroxyl group, and the net negative charge of the oxygen in that hydroxyl group, for example. But you can also have hydrogen bonds between amine groups, for example, or between an ionic compound and oxygen. So a positive ion and the negative oxygen, or a negative ion and the positive hydrogen in water. So hydrogen bonding is a weak dipole interaction for our purposes. Hydrophobic interactions are interactions between non-polar substances. Typically for enzymes and proteins, these are the reactions that are, these are the actions or the intermolecular forces that are forcing non-polar R groups inward in an aqueous solution, inward onto the inside of a protein. Now, I need to make a statement about some common mistakes. Complementary is not the same as matching. They are absolutely not the same. Complementary would be when you have this matches with this. All right, that would be complementary. They do not match. Match would be if they were both squares. These match. These are complementary. So these are complementary. These are matching. All right? So this will fit inside of here. These match. Reactants are not the same as substrates. A reactant may not be a substrate. A reactant may not bind to the active site of an enzyme. A reactant may just be a participant in the reaction, but the substrate is specifically a molecule that binds to the active site of the enzyme as part of this chemical reaction that is being catalyzed. So a substrate must bind to the enzyme. A reactant is not a substrate. A substrate may be a reactant, but a reactant is not always a substrate. They are not always the same thing. Now, word roots. Mono is one in Greek. Uni is one in Latin. Poly is many in Greek. Milli is 1,000th in Greek? No, Latin. Kilo is 1,000 in Latin. Intra is inside in Latin. Endo is inside or within in Greek. Extra is outside or without in Latin, and exo is outside or without in Greek. These word roots and prefixes and suffixes will come up as we continue forward. So, enzymes are globular proteins that catalyze chemical reactions. A standard chemical reaction, for example, would be carbonic anhydrase. That would be an enzyme. Carbon dioxide plus H2O yields carbonic acid, H2CO3. Or we could be building monomers into a macromolecule. DNA polymerase, building long chains of DNA, forming that phosphodiester bond. We could also be transferring groups, like for example, phosphotransferases. They transfer a phosphate group back and forth between molecules. Isomerases will change a molecule from one version to another. An isomerase will change alpha glucose to beta glucose or beta glucose to alpha glucose. Typically, we're talking about beta to alpha, though, just as a general rule. We also have enzymes that can digest larger molecules. Pepsin is a molecule that digests proteins in your stomach. Lipase is an enzyme that comes from your pancreas that goes into your small intestine. That digests lipids. Amylase is found in your saliva and your pancreas. Those are for the breakdown of complex carbohydrates. Now, I want to point out something. ACE, 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 ACE. ASE is a very common enzyme ending. If you see ASE at the end of a word, you're pretty much assured that it's going to be an enzyme. You're pretty much assured that it's an enzyme, which means it's made of protein, which means it's made of amino acid. Don't be fooled by questions that tell you, amylase, it's a carbohydrate, right? It's not. It's an enzyme. It's made of amino acids. All right. So, yeah. So, enzymes will catalyze chemical processes, which means they lower the activation energy by a reaction by participating without being consumed. Different enzymes do different jobs, because enzymes will only have one job. If a reaction can be catalyzed in one direction, then it's likely that that reaction can be catalyzed in the other direction, either by the same enzyme doing its job backwards, or by a different enzyme doing the opposite job. So, enzymes are produced within the cell because they are proteins. Therefore, they are made by translation. We can divide them into two groups by location of where they do their job. We have intracellular and extracellular. These terms will mean intracellular, within the cell, and extracellular, outside the cell. These enzymes, the ones that will work inside the cell, will be doing enzymatic reactions inside the cell and doing metabolic reactions and such. Extracellular reactions will be taking place outside of the cell, and I'll leave it to you to come up with examples on your own. This is the internet, after all. So, let's talk for a moment about activation energy. You need to start a reaction. You need to start the reaction by introducing activation energy. Activation energy is the hump that you need to get over to start rolling down that hill. So this is your activation energy in an endothermic reaction, and this is your activation energy in an exothermic reaction. You still need to input energy, no matter what type of reaction, whether you are eventually going to absorb heat or give off heat from the reaction, you still need to input energy in the first place to get that reaction started. It doesn't matter if you have one of those heat pads that you shake up or one of those cold packs that you shake up. You still need to do that activation energy of shaking up the pack. And that is going to start your chemical reaction. Your activation energy is shaking up the cold pack or shaking up the heat pack. And that is going to start your activation, and that is going to input your activation energy, start your chemical reaction, and you will have either endothermic or exothermic reaction. So, you have the energy of your reactants here. Your activation energy is applied. Your energy of your products is above the energy of your reactants. You are absorbing energy. That means the environment around your reaction is going to lose heat. Heat will enter into the reactants to form the products. You will lose temperature, heat in the form of losing temperature, outside the reaction, cold pack. Exothermic reaction. You have your energy of your reactants here, you apply your activation energy, and your energy of your products is lower than your energy of your reactants. You have this much energy to be released. That energy will be released to the environment in the form of heat, and that will make things warmer. This is your heat pack. Endothermic reaction, shaking up your cold pack and getting your cold pack. Exothermic reaction, shaking up your hot pack to get your hot pack, your heating. When you catalyze a reaction, you are going to take your activation energy and drop it down. So your activation energy of your uncatalyzed reaction is here, your catalyzed reaction is here. So, the energy of your reactants is the same. The energy of your products is the same. Nothing else changes. The only difference is how much energy do you need to put into the system to get your products. How much energy needs to go in before you get your products. So, this is your net energy, the unchanged. The net energy of your reaction is the same. Your reactants, your products minus your reactants is your net energy. That remains unchanged. Now, what the heck is this? This would indicate that there are multiple enzymes catalyzing multiple steps in one reaction. Even if you were to take glucose, for example, and burn it in a fire, that is one big step. Lighting it on fire is the activation energy, and you get carbon dioxide and water as your products. The net energy of reaction is energy of reactants, energy of products, energy released is here. So you release heat and light, and that is your release of stuff. So let's say we're burning glucose. So burning glucose requires one big step with a match, or multiple smaller steps with enzymes to get the same product. Heat and energy. Heat and energy in your chemical system. a chemical reaction. Carbon dioxide, water, and energy. So our first step, our catalyzed reaction has a lowered activation energy to here. Our second step has a lowered activation energy to here. Our third step has a lowered activation energy to here. Our fourth step has a lowered activation energy to here. And then we can proceed to products. Our highest activation energy, our third step is our highest activation energy. It didn't have to be the third step, it just was. If our second step had a higher activation energy, it would be that one. Whichever step has the highest activation energy, that will be our activation energy with our catalyst. That will be the thing that we have to get over. That will be our minimum required activation energy for our reaction to proceed catalyzed. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. So if we don't have enough activation energy to get past this point, we cannot proceed. If we don't have enough activation energy to get past this point, we cannot proceed. If we have enough activation energy to get past this point, we can proceed. If we have enough to get past this point, we can get past this point for sure. But if our enzyme is broken here, then we have this activation energy to contend with in the fourth step. We have a problem. All right? Now, the mode of action of enzymes. The mode of action of enzymes, the enzyme will bind the substrate to its active site, forming an enzyme-substrate complex. The chemical reaction, coaxed into taking place by the forces within the complex, cause both to undergo a change in shape. Afterwards, the product can be released by the enzyme, and it can return to its original shape, it being the enzyme. So this is an example of an enzyme causing the breakdown of its substrate. But it could just as easily be the reverse. We could just as easily have two substrates that are being combined into one product. So this is an example of an enzyme here. We have our one substrate and our one enzyme. These are complementary. This fits into here perfectly. This is our lock and key model. Right? So our active site and our substrate, one fits into the other perfectly. They bind together. That phrasing is very important. Binding. Not bonding, binding. And we have our enzyme-substrate complex. Enzyme-substrate complex. The enzyme-substrate complex, because there are new intermolecular forces at work here, the enzyme will change its shape, and the substrate will undergo stress. That stress is mechanical energy. That mechanical stress is going to cause product formation or separation into products, or the reaction to proceed in some way, the lowering of the activation energy. If this were two amino acids, it would be pushing the carboxylic acid group and the amino group into just the right position so this amine group and this hydroxyl group are right there so water can be ejected. So twisting of these substrates into the perfect position forms our new product. Now that our product is formed, it's no longer the substrate. We have different intermolecular forces. And now it will be ejected. And now the enzyme